Hello everyone. Our lab group was assigned the heterotrophic plate count procedure for this water microbiology lab experiment.
To understand the results of this procedure, some background information must be taken into account. First off, what even are heterotrophs? Heterotrophs are a group of living microorganisms, such as yeast, molds, and bacteria, that use organic carbon as food. Heterotrophic plate count, or HPC, is a method used to gauge the number of said heterotrophs in the water, since this number has implications about the quality of the water. A high HPC implies an environment that is easy for the growth of bacteria, while a low HPC indicates a difficult environment for the growth of bacteria. This is a necessary procedure since it can be used to test the water of recreational areas like pools and lakes, as well as drinking water sources, to determine whether they are safe for human use. HPC involves a counting of CFUs, or colony-forming units, and a CFU can form from a single cell, pair, chain, or cluster. During HPC, the CFU present on an agar plate is multiplied by the dilution factor and divided by the volume of the sample to give an estimate of the density of culturable heterotrophic bacteria per milliliter. In potable water, HPC levels should be less than 500 colony-forming units per milliliter. HPC levels may fluctuate, but counts that consistently exceed this value indicate poor water quality.
The materials used for this procedure was 6 buckets of pond water, 3 from Bigner Pond and 3 from Yucky Pond, 9 agar plate samples, a flame torch, 80% ethanol, 1 ml pipettes, 3 test tubes with 9 ml of sterile water, R2A agar, and a glass spreader. An R2A agar is a culture medium developed to study bacteria from potable water in a laboratory setting. This bacteria tend to be slow-growing species and would quickly be suppressed by fast-growing species on a richer culture medium.
First off, it should be noted that for this procedure, we stopped at a 1 to 100 dilution and didn't continue to reach a 1 to 10,000 dilution as the illustration represents. The first step performed in the pipetting process was obtaining 1 milliliter of our sample from the bucket, whether that be from the Bigner Pond or the Yucky Pond, and pipetting that into the first test tube, filled with 9 milliliters of sterile water, giving us a 1 to 10 dilution after proper mixing. We then extracted another 1 milliliter from this test tube with a new pipette and put it into our other test tube with 9 milliliters of sterile water, resulting in a 1 to 100 dilution, leaving us with a 1 to 10 dilution, 1 to 100 dilution test tube, and our 1x sample.
Step 1 of HPC. Obtain 1 milliliters from the sample bucket of pond water to the test tube marked as 1x. Then pipette 1 milliliter of the test tube 1x and into the test tube with 9 milliliters of sterile water labeled as 10 to the power of -1. Step 2 of HPC. Collect 1 milliliter of the test tube marked 10 to the power of -1 and use the pipette to mix the solution well into a clean test tube with 9 milliliters of sterile water labeled as 10 to the power of -2.
In the inoculation process, it is important to sterilize the plate spreader between every inoculation to avoid cross-contamination and errors in results. When inoculating, the plate spreader is dipped into 80% ethanol and passed through a flame for sterilization. For each plate, a 0.2 milliliter sample is pipetted onto the plate and spread evenly across using the sterilized plate spreader. The plates range in concentrations from 1x, not diluted, to x10 to the -1st, tenfold dilution, and then to x10 to the -2, which is 100-fold dilution. 3 samples were taken from each group named A, B, and C, respectively. After all the plates from each sample were inoculated, the plates were set aside to grow at room temperature for a total incubation period of 7 days.
To choose which results to count after incubation, we look for plates that presented between 30 and 300 CFUs, because this range is considered statistically significant. Results with fewer than 30 CFUs, as seen in Image A, can be skewed by small dilution errors in the presence of contaminants. If there are more than 300 CFUs, as seen in Image B, then there will likely be poor isolation as some colonies may have grown together. Image C shows the presence of a spreader colony that has overtaken the majority of the plate, and also shows the technique of using a Sharpie to mark the individual colonies as they are counted.
For sample B1, I chose plates A, B, and C for 1x dilution because they showed a desired number of colonies, 30 to 300 CFU. On plate A, there was 84, plate B had 70, and plate C had 77 colonies. In order to correctly represent these counts according to the dilution factor 1x and pipette amount 0.2 ml, I calculated the final numbers to be 420 for A, 350 for B, and 385 for C. The mean colony count for my plates is 385, and the standard deviation is 35. The small standard deviation suggests that there is little variability between samples.
For sample B2, plates A, B, and C for 10 to the power of -1 dilution showed numerous colonies. Plate A had 98 colonies, plate B produced 67 colonies, plate C produced 164 colonies. Plate C appears to have the highest colonies of all three plates. According to the dilution factor 10 to the power of 1 and the pipette amount 0.2 milliliters, I calculated 4,900 for plate A, 3,350 for plate B, and 8,200 for plate C. The mean colony count for my plates is 5,483 and 33. The standard deviation is 2,477.
For sample B3, I chose plates A, B, and C for x10 to the -1 dilution because they showed a desired number of colonies. On plate A, I had 26 colonies, and plate B, I had 38 colonies, and for plate C, I had 40 colonies. In order to correctly represent these counts according to the dilution factor of x10 to the -1st and pipette amount of 0.2 milliliters, I calculated the final numbers to be 1,300 for A, 1,900 for B, and 2,000 for C. The mean colony count for my plates is 1,733.33, and the standard deviation is 378.594. The large standard deviation number suggests that there is great variability between samples.
For sample Y1, I chose plates A, B, and C for 1x dilution because they showed the closest values to the desired number of colonies. On plate A, I had 26 colonies, for plate B, I had 22 colonies, and for plate C, I had 19 colonies. In order to correctly represent these counts according to the dilution factor 1x and pipette amount of 0.2 milliliters, I calculated the final numbers to be 130 for A, 110 for B, and 95 for C. The mean colony count for my plates is 111.67 and the standard deviation is 17.56. The small standard deviation suggests that there is little variability between samples.
For sample Y2, I chose plates of the 1x dilution because this was the dilution factor that presented the highest number of colonies, despite still falling outside of the 30 to 300 CFU range. On plate A, I had 23 colonies, on plate B, I had 18 colonies, and on plate C, I had 13 colonies. In order to correctly represent these counts according to the pipetted sample amount of 0.2 milliliters, I divided each plate's colony count by 0.2 and calculated the final bacteria density per milliliter numbers to be 115 for A, 90 for B, and 65 for C. The mean colony count for my plates per milliliter is 90, and the standard deviation is 25. The standard deviation is not so high, which suggests that there is not much variability between the samples.
For sample Y3, I chose plates A, B, and C for 1x dilution because they showed a desired number of colonies between 30 to 300 CFU. On plate A, I had 11 colonies. Plate B, I counted 48 colonies, and for plate C, I had 8 colonies. In order to correctly represent these counts according to the dilution factor 1x and pipette amount 0.2 milliliters, I divided each plate's colonies count by 0.2 and calculated the final bacteria density per milliliter. My numbers were 55 for A, 240 for B, and 40 for C. The mean colony count for my plates is 111.67, and the standard deviation is 111.39. The large standard deviation suggests that there is a great variability between samples.
Overall, the Bigner Pond showed greater concentrations in colonies on average as compared to the Yuck Pond. Bigner Pond might have conditions that are more suitable for bacterial growth than the Yuck Pond. Although some of the samples from the Yuck Pond had actinomyces present spreaders, the antibiotic properties may have prevented bacterial growth, lowering the total CFUs per pond Y. Plates Y2, 1xB, and Y3, 1xC showed the presence of spreaders.
For this graph, we are comparing the average bacteria densities for each pond sample. We can observe the Bigner pond sample's average densities seem to have a bigger abundance of bacteria per milliliter than the averages of yuck pond samples. We can visually see the differences between the two ponds and observe how B2 has the highest density of culturable heterotrophic bacteria per milliliter. This might be because Bigner pond may have conditions that are more suitable for bacterial growth than the yuck pond.
For this graph, the average bacterial density per milliliter between pond B and Y are being compared. Pond B showed a significantly higher average bacterial density than pond Y. Since potable water should have HBC levels of less than 500 CFU per milliliter, it appears as though pond B water would have to be treated to be safe to drink, while pond Y water appears to be within the acceptable HBC range.
Finally, we took a t-test. This test is applied to compare whether the average difference between our two groups, the ponds, has a real significance or not. These two independent groups will be compared to recognize the difference in means. When looking at the t-test paired to sample for means, we can see that the p2 tail is greater than the p-value, which is 0.05, which means there's no significance.